Hello, everyone, and thank you so much for tuning in to my talk. My name is Dr. Britt FLORKIEWICZ. I'm an assistant professor in the Department of Psychology at Lyon College, which is located in Batesville, Arkansas, in the United States. I am a primatologist. Most of my research is on the facial signaling capabilities of non-human primates. A lot of my research has focused on chimpanzees and hylobatids. These are two very distinct ape species that vary a lot in terms of their social and ecological environments. So today I want to showcase some of the research I've been doing in my lab here that illustrates just how differences in socio-ecological environments can actually result in differences in their ability to communicate using facial signals. So, before we begin talking about the research, a very brief overview of these two different types of apes. Chimpanzees live in incredibly large social groups. Sometimes it's a dozen or two individuals, but in some cases it can be over 200 individuals. They live in social hierarchies and they also exhibit evidence for fission-fusion dynamics. And they engage in a host of different social behaviors, such as play, using extractive foraging and tool use, and also engaging in cooperative hunting strategies. In contrast, we see that hylobatids, they tend to live in relatively smaller social groups of only a couple adult individuals and their offspring in a larger piece of territory. And we notice that they tend to coordinate their behavior through synchrony, and they're also well known for their vocal duetting, which they use to strengthen their pair bonds, but also to defend their territories from other neighboring hylobatid groups. So we're seeing that even though both of these are apes, they're varying quite a bit in terms of their social environments and also their ecological environments. One of these is an African ape, and the other one is a Southeast Asian ape as well. Now, recent studies coming out of my lab have revealed that there are also differences among these two apes based on their facial signaling capabilities. We're noticing differences in their facial signaling repertoire size and complexity, their ability to produce facial gestures, and also the form and function of facial mimicry. So I want to go over a couple of these studies today and provide some insight in terms of how we might be able to use these findings to make predictions about other species and expand this work to include other non-human primates. A lot of this research is being done in my animal behavior and cognition lab here at Lyon College. I've established this lab to help undergraduate students and also graduate students get research experience in animal behavior. So a lot of the studies you're about to see today have heavy undergraduate and graduate student involvement, and this is also open to international students and collaborators as well. It's the case where whenever we're working with multiple species and hundreds of hours of video footage, collaboration is going to be key in this regard in terms of getting those data, but also coding and analyzing those data. So. Before we begin talking about specific studies, you're going to notice that a lot of this work involves the use of facial action coding systems. These are systematic and standardized protocols for identifying all subtle and overt facial muscle movements. And it's a combination of these movements that creates a facial signal. So this is a very time-intensive coding process. It often takes hundreds of hours to get certified and to pass training tests to use FACS, but also to apply it to video footage for coding purposes. But there's a lot of benefits about using facts-based perspectives. So the traditional way of thinking about facial signals being produced by primates is, using an ethogram, where you're looking for specific kinds of facial muscle movements and specific contexts. So one of the most widely studied facial signals among primates is the play face. So this is where the corners of the lips are drawn backwards, the jaw is relaxed, and the lower lip is also relaxed away from the gum line. And the idea is that producing this signal means that you're down to play with other conspecifics. But the reality is that there's many different kinds of play faces that chimpanzees and other primates can produce, and they might vary in terms of social function. So when you're using facts-based coding perspectives like so, and you see here that underneath each image, there's a numerical combination, and this is using facts, where each facial muscle movement is assigned a unique code, and it's a combination of these codes that creates a signal. By using facts, we can get a better understanding of the nuances involved in producing this play face, but also when variation is likely to occur. And it allows us to study whether or not social variables, such as the intensity and duration of play, are related to these morphological changes in the display. So let's talk first about our comparative study that we did recently with chimpanzees and hylobatids. So what we did here is we applied facts-based coding to hundreds of hours of video footage on a host of different communicative events involving chimpanzees and hylobatids. We looked at facial muscle movements that both apes are capable of producing, and we wanted to see whether or not there's differences in their repertoire size and complexity. So, is it the case that one of these apes is producing many more different types of facial signals and maybe more complex facial signals involving more facial muscle movements than the other? And what we found was pretty interesting. We found that we were able to observe 65 distinct facial muscle movement combinations thanks to facts-based coding approaches. 21 of these are found in both apes, but only 14 are exclusive to hylobatids and 30 are exclusive to chimpanzees. Now, again, we're just looking at muscle movements that both apes are capable of producing, and it seems like chimps have larger facial signaling repertoires. When accounting for facial muscle movements that only chimps or only hylobatids are capable of producing, we see the same pattern that chimps are producing many more distinct facial signals compared to hylobatids, despite having only 156 recording hours compared to over 200 recording hours in hylobatids. We also noticed that chimpanzee facial signals, on average, are comprised of a greater number of facial muscle movements compared to hylobatids, and that's what this chart on the bottom left-hand side is representing. So we're seeing that chimps live in pretty large, complex social groups, and they also have very large, complex facial signaling repertoires. So there might potentially be a link between social organization and facial signaling capabilities. Now, it's not just about the size of the repertoire, but it's also about how you're utilizing this repertoire. So we're also doing a lot of research on something called facial mimicry. And this refers to the phenomena where an individual views the facial signal of a conspecific and produces facial muscle movements in response, usually produces the same types of movements, either in part or in full. So facial mimicry is often established based on two dimensions. It's based on how quickly you're mimicking those facial muscle movements and whether or not you're mimicking part or all of those movements. So in the research literature, you have rapid facial mimicry, which is producing those facial muscle movements within 1 second of viewing. You can also have delayed facial mimicry, where you're producing facial muscle movements within 2 to 5 seconds of viewing. And also you can think about this in terms of general trends and patterns between individuals within a given study period, producing the same types of facial signals. Now, we did a study in 2018 involving facial synchrony in hylobatids, so their tendency within a given pair group to produce the same types of facial muscle movement combinations. It's important to note that we're looking at instances of exact mimicry here, where upon viewing a facial signal, they're producing the same exact facial muscle movements in response. But in terms of the timing, we're thinking across the whole study period, right? So the tendency to kind of produce the same types of facial muscle movement combinations. And we notice an interesting relationship here that instances of facial synchrony increase, as does pair bond strength. So this implies that maybe facial synchrony is important either in terms of establishing or helping with maintenance of pair bonds. And the idea is that more facial synchrony provides greater pair bond benefits. So we're noticing that there's again a relationship here between how hylobatids are using facial muscle movement combinations during communication and their social organization. Now, we also need to think not just about the timing of facial mimicry, but the types of facial mimicry. So again, you can have instances where all facial muscle movements observed are being replicated, but you can also have type mimicry where maybe only a part of those facial muscle movements are being replicated within a given time frame. So we noticed that in the research literature, a lot of people tend to focus on one versus the other, or they tend to think of these as discrete phenomena. But the reality is this actually represents a continuum where you can either have exact or no mimicry, and then in between, you can have different levels of type mimicry. So my colleagues and I came up with a new set of measures that helps to kind of encompass this variation. And what you're going to see in the next study is we're looking at this in terms of RM or rapid mimicry size and RM or rapid mimicry ratio. And we did this study on specifically rapid facial mimicry, but you can use these measures for delayed facial mimicry and also facial synchrony as well. So with RM size, we're looking at the number of facial muscle movements that are produced within that 1 second window. And with RM ratio, we're looking at the proportion because as we just saw in the previous 2023 study, there's variation in the number of facial muscle movements within a given combination. So the proportion also matters. An RM size or RM ratio score of 0 means there's no RM whatsoever present. A score of 1 means exact rapid mimicry in this case is present, and values between 0.01 and 0.99 mean that type rapid mimicry is present to varying degrees. So we're able to use these measures to look at a host of different social variables, such as differences in social context. So you can see this on the next slide. This is a 2025 study that we're currently producing. It's under review and just got a minor revise and resubmit. And this study looks at the prevalence of facial mimicry across different social contexts in chimps. A lot of the research on facial mimicry has been done in the context of play. Play can be very risky, so it makes sense that communicative mechanisms that ease and facilitate the play process are going to be selected for. But what this study has found that chimpanzees are exhibiting evidence for facial mimicry across a host of different contexts. There's no significant differences in RM size and RM ratio scores across these 10 distinct contexts that were being observed. So this kind of implies that chimpanzees live in really large social groups. They're managing a host of different social relationships and they're managing a bunch of different types of social activities. So maybe facial mimicry has expanded to be encompassed in other social contexts to ease these types of complex interactions that are taking place. Now, another thing that we want to look at is the ability to produce something called facial gestures. So, when you're watching chimpanzees communicate, for example, they produce not just facial signals, but manual gestures, movements of the hands that are produced in an intentional, flexible manner in order to achieve a goal. So, for example, you see that this chimpanzee is producing both a piant hoot and a reach gesture, maybe with the goal of getting a piece of food from another chimpanzee. So, a lot of research on manual gesturing has focused on the hands, as the name implies, but we were curious to see whether or not hylobatids and chimpanzees are capable of producing facial gestures. And the idea is that maybe there's differences between these two apes because of the differences in their social environments. We're already seeing that hylobatids use more facial synchrony and chimps use more rapid facial mimicry and delayed facial mimicry. So maybe we're seeing that other differences are kind of spanning out based on gesturing capabilities. Okay. Another important thing to note is that we kind of expect that both of these apes are going to produce facial gestures to some extent because their hands aren't always going to be free because of locomotion. So let me first show you the results of our 2021 study with chimpanzees. We compared and contrast the 4 key gesture properties and their associated variables among chimpanzee facial signals and also their manual gestures. And we noticed that excluding one variable related to intentionality, all other variables observed exhibit consistent patterns between their facial signals and also their manual gestures, which kind of implies that there's not a lot of differences here, that facial signals are in fact capable of being used as gestures because they're exhibiting evidence for communicativeness, intentionality, flexibility, and goal association, just as their manual gestures. And it's in very similar proportions as well. So we see that chimps do produce facial gestures in this regard. Now, we also decided to extend this to hylobatids, the 4 different species. that we included in our previous studies. And what we found was something really interesting. Now, again, we're comparing, in this case, hylobatid facial signals to the chimpanzee facial signals in terms of the four key gesture properties and the associated variables that have been studied extensively by the other researchers before. And we're noticing a combination of consistent and inconsistent patterns here. So take, for example, we see consistency with flexibility. So those two variables, persistence and elaboration, they're occurring significantly above chance in both chimps and hylobatids, but notice that a lot of variables associated with goal-oriented behavior and intentionality, not so much, unfortunately. So we're seeing that, hylobatid facial signals, they're communicative and flexible, but they're not really intentional or goal oriented. And again, maybe this is because of the fact that hylobatids live in very unique social environments compared to chimpanzees. They may not need to produce intentional or goal oriented signals if other mechanisms like facial synchrony are already in place to do a lot of the heavy lifting. Synchrony helps to better understand what your partner might be thinking or experiencing, and it allows you to better predict their behavior. So maybe you don't need these variables of intentionality and goal association because they're doing facial synchrony and also other forms of synchrony that we talked about, such as vocal duetting. So, to kind of conclude, what this research has shown is that chimpanzees and hylobatids, they live in socially and ecologically distinct environments. And because of these differences, maybe we're starting to see differences in their facial signaling capabilities. Chimps have very large and complex facial signaling repertoires. They exhibit evidence for rapid facial mimicry and delayed facial mimicry. And we're thinking about exact and type mimicry. Chimpanzee or hylobatids, excuse me, we do see evidence for facial synchrony, specifically exact facial synchrony, where members of a given pair are producing the same exact types of facial muscle movement combinations at similar rates. But we don't have as strong evidence for rapid facial mimicry just yet in hylobatids. Now, additional research needs to be done, especially with hylobatids, because compared to chimps, there hasn't been a lot done on their socio-communicative capabilities other than vocal duetting and pair bond activities. So we need to extend this work to include facial signaling behavior more so than what has already been done. And the same needs to be done with chimpanzees. Clearly, we're seeing that, for example, rapid facial mimicry, it's not just play, it's occurring in a lot of other contexts. So we need to really extend out and do more studies with these two apes. It's also my goal that we can use these kinds of studies and their results to make predictions about other species. It seems that group size and also complexity in certain kinds of social activities might indicate that you're more likely to see larger, more complex facial signaling repertoires, more evidence of facial mimicry and vice versa, and also more evidence of facial gesturing. Now I want to apply this to other species with my students, right? There's dozens of other primate species that live in large social groups and exhibit evidence for communicative complexity and other modalities, and also do cognitively complex behaviors. So I'd like to ideally start expanding these types of studies to monkeys, and Japanese snow macaques are a great example of perhaps very large social groups and complex social organization. They also exhibit extensive variability in their behavior because they live in dramatically differing environments, all the way from the northern to southern regions of Japan. So it's quite possible that we're going to see not only evidence for facial gesturing, for large complex facial signaling repertoires in these macaques, in addition to evidence of facial mimicry, but maybe variation based on locality or region that they're occupying. So our hope is to expand this kind of work to include other species, in addition to continuing our research on the facial signaling capabilities of chimpanzees and hylobatids. Now, a lot of the studies I showed today, these were like the quick SparkNotes version of these, where I was only able to kind of cover a couple of key results, but all of these articles are available. If you're interested, please send me an email at brittany.florkiewicz@lyon edu, and I'd be more than happy to share copies of these studies. And also, if you are a collaborator that wants to get involved in some of these projects, or you're a student, again, we take international students, domestic students, and all you need to have is a curiosity and passion for animal behavior research and a little bit of time each week dedicated towards coding and analyzing video footage to be part of these types of studies. Thank you so much again for attending my talk. I greatly appreciate it. And if you have any questions or concerns, let me know. Always happy to chat more about primates. Thank you so much. Have a fantastic rest of your day. Bye-bye.